In today's video, we're going to be taking a look at synaptic transmission for A-level biology. Now, the first thing that we need to understand is what is a synapse? So a synapse is simply the point of communication between two different neurons or between a neuron and another type of a cell, like for example a gland or a muscle, right? In the example of a neuromuscular junction, it's a neuron connected to a muscle. Now, the way that the synaptic transmission works is by using chemicals that are called neurotransmitters. And a type of a neurotransmitter that we will actually be looking at today is acetylcholine. Now, where the synapse uses acetylcholine as the neurotransmitter, it is going to be called a cholinergic synapse. Now, in the synapse, we have three main features. We have the presynaptic neuron, we have the postsynaptic neuron, and in the space that is in between them, which is about 20 to 30 nanometers wide, is called the synaptic cleft. Now, in the presynaptic neuron, we have the presynaptic membrane. Now, we can see that at this axon terminal, we have this knob shape, and this is called the synaptic knob. Okay, so in the presynaptic neuron, we have the synaptic knob. Then in the postsynaptic neuron, we're going to have the postsynaptic membrane. What we can see here is we have synaptic vesicles, and the synaptic vesicles contain the neurotransmitter acetylcholine in this case. Now, it's very important to understand that synaptic transmission is unidirectional, and this is because neurotransmitters are used to pass on the signal. And because the neurotransmitters are only in synaptic vesicles, which are only found in the presynaptic neuron, any impulse that may be arriving to the postsynaptic neuron will simply stop at that point, because it will have no neurotransmitters to be able to send the signal to the presynaptic neuron. Okay, so it's unidirectional because those neurotransmitters are only found in the synaptic knob, which is part of the presynaptic neuron, right? Pre means before, so before the synapse. Post means after. Okay, so postsynaptic after, presynaptic before. Now let's take a look at how the synaptic transmission would actually work using the acetylcholine as an example. So we're going to have an action potential that is coming along the axon of the presynaptic neuron. That's going to depolarize the presynaptic membrane, causing voltage-gated calcium channels to open. So calcium ions will enter the synaptic knob, and when they do this, they're going to stimulate the synaptic vesicles to fuse with the presynaptic membrane, releasing the neurotransmitters into the synaptic cleft. Okay, so we've had the calcium ions have entered because of the action potential that came along the axon in the presynaptic neuron. Those calcium ions then caused the synaptic vesicles to fuse with the presynaptic membrane to release the neurotransmitter acetylcholine into the synaptic cleft. Now what we have is the neurotransmitter, the acetylcholine will bind, the acetylcholine molecules will bind to the ligand-gated sodium channels, which are closed at the moment. Now, 2 of 5 protein subunits that make up these ligand-gated sodium channels must be bound to by an acetylcholine molecule. And when this occurs, the ligand-gated sodium channels will open, and this will enable sodium ions from within the synaptic cleft to enter into the postsynaptic membrane. So they will go through the postsynaptic membrane. This will cause excitatory postsynaptic potential. So before, in one of the previous videos, we saw that the generator potential in sensory neurons occurs when we build up towards the threshold value from the resting potential. In the postsynaptic membranes, so in the postsynaptic neurons, this is instead called an excitatory postsynaptic potential. So the more sodium ions that enter, the more excitatory postsynaptic potentials will be generated, and if enough are generated in quick succession and the threshold value is reached, what will then happen is an action potential will be stimulated in the postsynaptic membrane, and that impulse will carry along in the neuron. Now, it's important also to understand that this could be with a sensory neuron going to a motor neuron, or it could be with a sensory neuron passing its signal on to an intermediate neuron, and then the intermediate neuron would pass that signal on to a motor neuron, right? These are different examples. Now, beside the ligand-gated sodium channels are going to be enzyme-bound molecules called acetylcholinesterase that break down the neurotransmitter acetylcholine into acetate and choline. Now, the importance of this is so that the sodium ion, so that the ligand-gated sodium channels don't remain open constantly, and they only remain open when there is that nerve impulse coming through in order to continue the action potential in the postsynaptic neuron. Okay, because if the acetylcholine wasn't broken down, then the action potentials would be constantly stimulated in that postsynaptic neuron. So now we've got the acetylcholine broken, like it's been hydrolyzed by acetylcholinesterase into acetate and choline. What then happens is the acetate and choline go back into the presynaptic neuron, so in the synaptic knob, and together with the help of ATP that is produced by the mitochondria in the synaptic knob, they will be recombined into acetylcholine, and they will be stored in the synaptic vesicles, ready for use for the next impulse, for the transmission of the next impulse that may come along in that presynaptic neuron. That's it for this video. If you found it helpful, take a look at some of the other content on my YouTube channel that might interest you. Otherwise, thanks very much for watching, and I hope you have a great day. Bye-bye.